Hey friends, welcome back to another lecture session regarding DNA structures. We might know that there are three forms of DNA we study. Those are A, B, and Z forms. Let's begin. Today I have collected all the points or features for all the three types of DNA. These are frequently asked areas or important areas for all types of examinations, or maybe your MCQs, any type of examinations related to biology or life sciences. This area is important in making questions. So first features, here you can see how I have tabulated DNA A, DNA B and DNA Z, and on the left hand side column it is written as features. So what are the features of these DNA forms and what are their, we can say, the exact values for the features. Now the first is height. Height is considered from its imaginary axis to the helix height they are talking about. The DNA is a double stranded helix, and the height of each helix from the imaginary axis of the DNA. For DNA A, it is 24 angstrom. For DNA B, it is 34, and for DNA Z, it is 44. So it is very easy to remember. We have to remember these points for solving questions in the examination. So height 24 angstrom, 34 angstrom and 44 angstrom for Z. Okay. Next is base pair per helix. So how many base pairs are present in a single helix that they are talking about? So it is 11 base pair in A form of DNA. There are 10.5 base pairs. Okay. 0.5 वाला जो है part वो next helix में आ जाता है 0.5 वाला, इसलिए 10.5 they have taken. Then Z form में there are 12 base pairs. Okay, next feature comes diameter. Okay, again they are considering the diameter of the helix from the axis, so that is 28 angstrom, 20 angstrom and 18 angstrom. So likewise for A form 25, for B form 20 and for Z form 18 angstrom. So these 3 points are frequently asked in the examination. Next we will see the helicity. So how they are moving, the helicity of the helix. Okay, so this is a right-handed A form of DNA that the helix moves in a right hand direction. Okay, and for B form of DNA it is left-handed, and for Z form also it is left-handed. So only the A form is right-handed. Next is major groove. Major groove, narrow and deep. So in DNA helix, if I draw here over here the structure of DNA, you might observe one thing, that if this is a DNA structure, we have to always draw DNA structure where the strands are parallel to each other. Okay, try to draw the structure where the strands are, most of the time it has to be parallel. Okay, so here these strands are parallel. Now the next is a major groove. So major groove, these grooves are major groove. Let me tell you, let me show you in the diagram. So these are major grooves. This is major groove. Groove means a place which shows a cleavage kind of area or a furrow kind of space. So this is a major groove. Again, this is a major groove. And the minor grooves are, so these are the minor grooves. Okay, where angle, like you can see the angle or space which are less. So that is minor groove. Okay, these are minor groove. And now you know what is the major groove and minor groove. Next, let's see the next point, sugar puckering. So in chemistry, you might have learned in 11th and 12th standard, any structure, any chemical structure, when we observe, we understand its puckering. Okay, there is a term called sugar puckering, okay, or the structural puckering in any structure. So in case of DNA A, it is 2' exo and 3' endo. In DNA B, it is 3' exo and 2' endo. How to remember? Just shuffle it. 2' exo, 3' endo. 3' exo, 2' endo. Okay. Then for D form, you have to remember for both purines as well as pyrimidines. So that is for purines, it is same as for DNA A. You can observe here. So remember like that. For purines, we have the feature of same as DNA A, and for pyrimidines we have same as for DNA B. Okay, easy. Yeah, for major and minor group, we have left something. For DNA A, it is narrow and deep. So the groove is for DNA A, it is narrow and deep, the major groove. While in case of DNA B, it is wide and intermediate, somewhat wide for DNA B form, and for Z, it is flat. Okay, minor. In case of minor group, for DNA A, it is wide, then for DNA B, it is narrow and intermediate. So just remember, DNA B is the most common form seen in living organism. The major groove in DNA A, sorry, in B, the major groove is wide and intermediate, while the minor groove is narrow and intermediate. So just you have to remember two terms, wide and narrow. Okay, wide and narrow intermediates. Then for Z form, it is narrow and very, very deep. Okay, this was left somehow by me, so sorry for that. Let's move to the next point, glycosidic bond. uh, Again, the conformation, the glycosidic bond conformation, there are terminologies in biochemistry or chemistry we have learned. The angle, so that we call this an anti, the glycosidic bond conformation is anti in A form of DNA, anti in B form of DNA, and in case of Z form, for purines it is syn, and for pyrimidines it is anti. Only we have to remember that in Z form of DNA, purines are syn. Rest all are anti forms for glycosidic bond. One more important term, that is helix rise per base pair. Okay, so what is the helix rise per base pair of DNA? So for A form, it is noted as 2.6 angstrom. For B, it is noted as 3.4 angstrom, and for Z, it is noted as 3.6 angstrom. So friends, whatever the basic points that we have already learnt in 11th, 12th, or in our BScs, okay, these are just the features. This is an important feature that is asked. Rest of the things, generally they don't ask in the examinations. Okay, for making MCQs type of questions. So these are very important points. If you learn them, so we can cover maximum areas regarding the nucleic acid polymorphism. We also call this as DNA polymorphism, or different structural form of, we can say, structural forms of DNA. Those are A, B, and Z. So let's move to the next part. One more slide we have for this topic. So I just wanted to give you a brief idea with the help of a diagram, that how A, B, and Z forms of DNA look like. Okay, let's move. So here in this diagram, you can see this is the A form of DNA, this is the B form of DNA, and this is the Z form of DNA. From your lower standards, lower classes, whatever you have seen the diagram of DNA in your textbook, you'll definitely uh, make an idea that this is the structure we have observed in our textbook. Yeah, that is the B form of DNA. While the A form and Z forms are not, we do not see them generally in our textbooks, because the A form of this form is generally favored by RNA, double stranded RNA. If it is there, then it favors A form structure, that is quite compact, है ना, थोड़ा सा ऐसा हम कह सकते compact है as compared to B or Z, है ना. Then B form, standard DNA double helix. Yes, helix under physiological conditions. So हमारे बॉडी में, लिविंग ऑर्गेनिजम्स के बॉडी में जो बायोलॉजिकल फॉर्म ऑफ डीएनए है, दैट इज बी फॉर्म ऑफ डीएनए, दैट इज स्टैंडर्ड डबल हेलिक्स है. देन Z फॉर्म ऑफ डीएनए है हमारे पास इन द थर्ड डायग्राम. This one is the Z form, and here you can see that this is a live laboratory anomaly. It means there is an abnormal form of DNA in lab conditions for studying purpose. Okay, if the DNA is treated with some chemicals, then this form of DNA is generally observed. Okay, for study purpose we consider all the three, and generally the features are the important. Okay, so I hope this is clear for all of you. Okay, and now the A, B, and Z forms of DNA are clear for all of us. Thank you for listening today's session. It was very short session, so if you uh, remember all the features clearly and understand them, it will be easy for your solving, like practicing questions and solving the questions. So if you have any queries or you want to share anything, you can mail me on this email ID, and this is the link here. You can write your experiences. So thank you so much. Let's see you in the next video session. Thank you.